Hello everyone. I know exam is just around the corner, and so we are continuing with our paper reviews. And so we are continuing with BGCSE Biology. And today we're going to do a paper 1, and this is from the year 2019. Now, always remember to write your school number, very important. Also remember to write your candidate number, your surname, and your initials. Always write those in. Again, remember to read through the instructions carefully and also follow the directives of the examiner. Now let's jump into our first question, and this question reads, which characteristics of living things occur when a butterfly sucks nectar from the nectary of a flower? And so the option A saying growth and reproduction cannot be correct because the organism is not growing. So anything with growth and reproduction, they are incorrect. So you can look at option C as well, include growth, and so those are not correct answer. So for the butterfly to reach the flower, the butterfly will have to move, and then the nectary that it sucks from the flower, it will use that for nutrition or for food. All right, so B will be our correct answer for number 1. Now, number 2 is that which is the correct way to write the scientific name for the yellow elder plant? And the yellow elder plant is the Tecoma stans. And now what is important here? All these words are saying the same thing, but the concept of writing scientific name is that you should always start the genus, which is the first part of the name, with an uppercase letter. So the answer could only be between A and B, and the species must be written with all lowercase letters. So therefore B will be the correct answer. All right, so let's now jump into question number 3. Question number 3 reads, which environmental factor is biotic? Biotic means living, okay? And the opposing word for that, which is non-living, is abiotic. So light intensity, pH, temperature, they are all non-living part of the environment. So predation, representing predators, will be the living part of the environment, okay? So let me write that word for you. Abiotic is the non-living part of the environment. Okay, just please remember that. All right, so let's go to the other question, number 4. And so question number 4 now reads that the diagram represents a food web. It says which organism in the food web is the tertiary consumer? And so let's now check first. The plant is a producer. So we have the producer here, which is a plant. That was kind of like that. And so let's start here. The grasshopper is the primary. Lizard is the secondary. And then the hawk will be tertiary in this case. Okay, going across from the grass as well, going to the rabbit. So the rabbit will be primary and also the hawk would be secondary if you go this way. So depending on the route you're taking, the hawk is the only one actually getting the position of a tertiary consumer. So you could have grass, you could have the mouse, and then you could have a snake, and then you could have the hawk. So in 2 ways there, we notice the hawk is the tertiary, so the hawk will be the correct answer here. All right, so let's go to the next question. And question number 5. So question number 5 now is that the picture shows an aquatic food chain. And in this aquatic food chain here, we have phytoplankton, we have zooplankton. And just to make it known right here that phytoplankton, they are plant-like, and zooplankton, they are animal-like, okay? And so the question here is asking now, it said at which trophic level is the tuna? So let's now look at where the tuna is. And so let's start from the producer, which is the phytoplankton. So trophic level 1. So this is 1, trophic level 2 for the zooplankton, and trophic level 3 for the mackerel. And so the tuna will be our trophic level number 4. Okay, so let's go to which option there is correct for this. So this is number 4. And so this answer is D. That's the 4th trophic level. And please remember that the trophic level is each level of the food chain or the food web starting from the producer. So the producer is always the first trophic level. after the producer, you will have your herbivore. Your herbivore is the first consumer. So the consumer level starts after the producer, but the trophic level starts from the producer. Okay, so please make a note of that, and please remember it and do not get them mixed up. All right, question number 6. Now, question number 6 is which is the correct zonation of mangroves in the Bahamas? I go ahead and write the scientific names for the 4 mangroves. And so we have the red mangroves, which is called the Rhizophora mangle, the black mangrove, which is called the Avicennia germinans, and the white mangrove, which is called the Lagunocularia racemosa, and the buttonwood, which is Conocarpus erectus. And so in this order listed on the side here, that's the order from the sea to the land. And so question number 6 here will be D, red, black, white, and buttonwood. Okay, so this will be D. Okay, and also need to know the features of these mangroves in terms of their root system, their leaves, and also their special adaptation in terms of where they are found. All right, so let's go to our next question here. Question number 7. And question number 7 reads that what adaptation of animals living on a sandy beach helps to protect them from drying out and predators? So there are 2 things you're considering right here. One is to prevent dehydration, drying out, okay? And also to be protected from predators. So which will be the most appropriate answer here? And so looking at option A, is burning in the sun, which is a good answer. Camouflaging may help them escape from predator or hide from predators, but will not protect them from the dehydration process, all right, or protect them from the sun. Exoskeleton, this might be quite handy in terms of drying out, but not necessarily protect them from predators. Okay, filter feeding is definitely not the answer. So A will be our best option right here, and it's simply to burrow in the sand. So they burn the sand to escape the direct sunlight and also to hide from predators. So that's a great answer right there. All right, so question number 8. Now, question number 8 is which apparatus is being used in the ecological field study? And so I make some special notes here for you, right, just to save some time. And so this apparatus is called a quadrat, all right, and so I'm going to talk about what these are just a minute. All right, just to point out something about a quadrat, right? A quadrat will be in squares. So let's say I put this in half here. There'll be equal squares and then going across like that, and then probably the next one right here. All right, so normally you see like strings or wires going across it like that. All right, and they're equally distributed or measured. Okay, mine may not be accurate, but just bear in mind that these squares will be equal in size. All right, and so this is a good way to study organisms. So probably you toss this in an area and you'll go and check the squares and see how many organisms are present, and then you can estimate how many organisms will be present in a particular area. All right, so that's a very good way to do a field study. Now, I make some special notes right here in terms of the different devices that they actually list there, or apparatus. So we have what they call a pitfall trap. And a pitfall trap is used to catch small organisms that are usually on the ground. And I have a diagram for that. I will show you at the bottom. Quadrat, which is shown in the diagram above, it is used to measure the distribution of organisms in an area or habitat. And then we have a sweep net. And a sweep net tend to be cone-like, or it will be funnel-like or funnel-shaped, and is used to capture insects or small organisms that are typically found on grasses or on the plant foliage or the leaves. So let me just point this out real quick. So for the pitfall trap, you'll see the diagram like this here. So it's pretty much you could put like a cone inside of the ground, or just dig the ground out and just cover it with rocks, or you could use it covered with a metal plate. So there are many ways which you could have a pitfall trap. There are many different designs or different ways to do it. Now, a sweep net, which is this on the right in blue, a sweep net, you pretty much will see this if you watch SpongeBob, right? SpongeBob and Patrick would be catching jellyfish with their sweep net, running and catching fish, jellyfish. So a sweep net, you move it back and forth in the grass area or where you have small shrubs. And if insects are on those grasses or the shrubs, then you'll catch a few of them. And so you can measure or count the amount that you catch, and then you estimate the possible amount that are present within that area. In agriculture, it is also used to capture insects as well that may be eating certain crops. All right, so it's a very good and useful thing to kind of measure the amount of organism in that particular area. All right, let's go to number 9. And so question number 9 is said, what is a natural threat to an ecosystem? So in terms of natural threat to any ecosystem are those things that are not caused by man. And so deforestation is an action caused by man, cutting down trees, especially more than what they can grow. And so the area will become barren. All right, and so, or lack of plants. And so disease is something that we cannot control, and that's a natural thing to have disease in an ecosystem. So number 9 will be B. All right, and so genetic engineering will be action of man. Overfishing is action of man as well. So disease is something that we cannot control. All right, number 10. It said which pollutant correctly matches the effect it has on the environment. And so going through the list here, carbon dioxide causes ozone depletion. No, carbon dioxide will cause greenhouse effect, which will lead to global warming. Chlorofluorocarbons, those could cause depletion of ozone. All right. And sulfur dioxide causes acid rain, which is good. So C will be your answer right there. Smoke as well will cause some respiratory problems. All right. And smoke will will release carbon monoxide or carbon dioxide as well, depending on what you're burning. All right. All right. So let's jump on to number 11. It said which water pollutant is commonly found at the Clifton Pier. Now, Clifton Pier is where you'll find BPL plant, okay? And so what they use to produce electricity is oil, and so what you most find out there is crude oil, if you should find any. Fertilizer, pesticides, and those will be on farm areas. So you find these on farm areas or areas that they do farming. Sewage could be where you have a lot of residential places or sites. All right, so generally we have a large population of houses and so on. That's where you'll find mainly sewage, or you may have large factories. All right, so number 12, it said which product is biodegradable? Biodegradable, as the word bio means living, degradable means to break down. And so remember what biodegradable means is to break down by living organisms such as decomposers. All right, and decomposers will include bacteria. And so plastic, metal, and glass, they are non-biodegradable. So the answer here will be D, which is wood, which is coming from plant. All right, so let's go to number 13. And question number 13 is said, what type of cell is shown in a diagram? Well, I've gone ahead and labeled this diagram. So we have the nucleus, the mitochondria, chloroplast, vacuole, cell wall, and cell membrane. And by virtue of the shape of the cell and also the presence of a cell wall and also chloroplast, then we know this must be a plant cell. All right, so plant cell will be our correct answer right there, which is option D. All right, for number 14, it said which of the following correctly matches an organelle with its function? And so we have central vacuole is a control cell activities is not true. The nucleus will do that. Chloroplast make glucose. That's sounding really, really good. So chloroplast is where photosynthesis take place and produce glucose. So let's highlight that for now. Mitochondria store water, which is not true. Mitochondria will produce energy. Nucleus store glucose is not true. Glucose control the cell activities. All right, and the central vacuole is what store water, plant food, all right, and waste. All right, so let's go to number 15 now. It said, which two organelles are present in plant cell but not in animal cells? All right, so cell membrane in option A is not correct because cell membrane is in all cell. Chloroplast is a correct choice. Cell wall and chloroplast, they are definitely the two things that are present in plant and not in animal cell. So we can stop right there. Okay. Cytoplasm is in all cell. Nucleus and cell membrane pretty much in all cells as well. All right, so let's jump on to number 16. Number 16 is how do water molecules move during the process of osmosis? And water molecules will move from dilute solution to concentrated solutions or from a hypotonic to a hypertonic solution. So let's see, we're moving from dilute to concentrated. So A is incorrect. So B, no, not B, because B said from concentrated. So those two will be totally incorrect. So C and D must be one of the answers. So let's look at C. Is it from dilute to a more concentrated solution? Definitely is correct. D is really not a good answer because saying from dilute to a more dilute is not true. And just to make a note of this, a dilute solution is classified as hypotonic solution, right? So hypotonic, if you especially comparing with the internal part of a cell. So hypotonic, and if a solution is concentration, then we can use the word hypertonic to describe that, okay? So hypertonic, just remember the words. And if the solution is equal in concentration as to what is in the cell, then you classify that as isotonic, okay? Just to make a note of that. All right, so great. Let's go to number 17 real quick. All right, so number 17 here, it said, what will happen to red blood cells, the red blood cell when placed in distilled water? All right. Now, for option A, it's a double in size, then stay that way, which it can double in size, which is true. But if it's distilled, it's going to keep on increasing size. All right, so a double in size can occur, which means getting bigger. The same shrink, the size shrink, which is B, is not correct. It stay the same, which is also not correct. So once it's placed in distilled water, which means it is in a hypotonic situation or solution, then the red blood cell will tend to swell, and then it will eventually burst, okay, because of a lack of cell wall. No cell wall to maintain the shape of the red blood cell at all, okay. Now let's go to question number 18. And so question number 18 now reads, it says when starch and amylase are mixed in the presence of iodine, all right? So just hold a minute right here. It says when starch and amylase are mixed. Remember now, iodine will change to blue-black in the presence of starch. But now amylase is there. So amylase is now going to play a key factor. Okay, so place amylase with the starch. All right, they say the blue-black color rapidly disappears. Okay, so that means the starch is reacting with the amylase to form something different. So the iodine cannot maintain its blue-black color. That means it's no longer starch. Okay, so this happens because the starch is turned into which of the following? So starch is being changed into something else. But remember now, amylase will break down starch into maltose, and this will take place in the mouth if you're comparing it to where in the body is taking place. So starch break down into maltose, and so maltose will be your answer right here. Now what I want to point out to you, maltose will eventually break down into glucose. Okay, so that's very important. Okay, maltose will eventually break down into glucose. So to point this out again, starch is a polysaccharide, maltose is a disaccharide, and glucose is a monosaccharide. Okay, and so glucose will be absorbed inside of the body. All right, so let's go to question number 19. All right, and so for question number 19, right, so let's go into it real quick. So question number 19, now we said use the graph below to answer question 19 and 20. Now, they say that the graph shows the relative rate of an enzyme control reaction at different temperatures. And so we have an increasing enzyme activity on the y-axis, and then we have temperatures on the x. It said, which describe the shape of the graph within the temperature range marked X. And so X is the highest point, which means there you have the maximum reaction or the optimal temperature. So here you have the maximum rate of reaction. So this will be our correct answer, option C. All right, so now let's jump back into our next question here now, and we're going into question number 20. Now, question number 20 said, at which temperature is this enzyme totally denatured? The word denature for enzyme represents that the enzyme changed, the shape of the enzyme has been changed because of unsuitable conditions. Okay, so temperature is one of them that could change the shape of enzyme. And so let's just go back to the graph and talk about when it's totally denatured. So again, maximum temperature is right here. Okay, and so at this point is when it is totally denatured. From here to here on the left going up, all right, this is where it is least active. All right, so at this temperature could be inactive, and then it start become activated, maximum reaction. Then at this point it will be totally denatured. That means the reaction will stop. In other words, the substrate will be unable to fit in that enzyme. So that will be about 60 something. So let's see which is the correct answer here. Answer number 20, yeah, the best answer here is 63. Okay. All right. Awesome. Now let's go to number 21. And so question number 21 now reads, it says, which gas is a waste product of respiration? Okay, and so respiration, the gas that will produce after respire is carbon dioxide. Okay, all right, so there's not much to talk about right there. All right, let's go to 22. It said, what is the product of anaerobic respiration in the muscle tissue? Now, what is important? You need to know what type of anaerobic respiration is taking place in the muscle tissue. Because remember now, there are two types of anaerobic reaction. Okay, so you have one that is called alcoholic fermentation, which is not taking place in the muscle. All right, that is done by yeast, for example, and that will produce alcohol or ethanol. All right, and carbon dioxide is also produced in that type of fermentation. The one that takes place in the muscle, especially during strenuous exercise, is called lactic acid fermentation. And so lactic acid is being produced. All right, and please, you need to know the reactions for these respiration processes. And so please, you could look at the lesson that I have on aerobic versus anaerobic respiration, and you will see the reactions and the amount of energy being produced. Let's rush into 23. Question number 23 said, which row in the table shows the correct percentage of oxygen in inhaled and exhaled air? Now remember that the oxygen coming in, which is inhaled, is anywhere between 20 to 21%. Okay, typically it's 21%. For the most part, you see 21. All right, now in exhaled air, it will be 16%. Okay, so answer here for 23 will be C. All right, so it is C is the correct answer. All right, so now let's jump into question number 24. Now, question number 24, it said that the cell that line the trachea have cilia. And so cilia, those are the hair-like structures. It said, what is the function of these cilia? So the cilia will actually sweep dust particles, especially when they combine with mucus. Okay, so our answer here will be D. Okay, our answer there will be D. Now 25, and we're halfway through. Okay, again, in the interest of time, I put some information here for you. So don't look at those things on the side yet. Just look at the options, and I talk about those things on the side. Do not be distracted by them. All right, as a matter of fact, let me put a line here, and again, I will discuss them after I answer the question, because the reason I put them here. And so the question reads now, in systemic circulation, blood is pumped to all parts of the body except which organ? Okay, so again, we pump all over the body. The only thing is not pumped to in systemic is the lungs, because the lungs will have its own type of circulation. Okay, and so hence the reason why I separate the different types of circulation on the right. And so systemic circulation is simply blood moving from the heart to the body back to the heart. That is systemic circulation. Okay, Now for pulmonary circulation, blood is, and remember the word pulmonary always refer to the lungs, so like the pulmonary artery, pulmonary vein, right? And so blood flows from the heart to the lungs and back to the heart. That is pulmonary circulation. Now in examination situation, you can also explain the blood vessels that are involved in this. Now let's say, for example, in systemic, blood moving from the heart to the body, it goes to the aorta. Coming back from the body to the heart, it comes through the what? Vena cava, which is the vein. Now in pulmonary circulation, blood flows from the heart through the pulmonary artery to the lungs, and then through the pulmonary vein and back to the heart. All right, and so we have a next type of circulation, which is called coronary circulation. So coronary circulation is that the blood will flow to the muscles of the heart. Now, why this is necessary? The heart itself requires blood. Why? Because the heart is a working muscle, and muscles need oxygen, need glucose and other nutrients. I'm going to talk about glucose and oxygen mainly. The purpose of glucose and oxygen is simply to produce energy by respiration. If the heart muscles are not getting enough energy, then they will slow down, and if no energy at all, then they will stop, and the person will have an heart attack. And of course, you know the consequence of that, right? So definitely the blood itself need to flow to the heart, so the heart can work by producing energy. All right, without energy, the heart will stop working. All right, so it's very important to have that. Okay, number 26. Now in question number 26, it said, in which of the following structures is blood pressure greatest? Now, right off the bat, you need to remember that arteries will have the greatest what? Pressure. Now, we have two arteries here. We have the aorta and we have the pulmonary artery. But let's decide which one will have the greatest pressure. The jugular vein, vena cava, those are two veins. But let's decide between the aorta and the pulmonary artery, which one has the greatest pressure. Now, the pulmonary artery, as we even mentioned above, only flows to the lungs. However, now the aorta will flow all over the body. And if it's going all over the body, it therefore means it requires a greater amount of what? Pressure. So 26 will be A. All right, let's go to 27. Now, question number 27, it said the diagram shows the human digestive system. And I have gone ahead and labeled this for you just to save some time. So L is the liver, M is the pancreas, O is the small intestine, and N is the large intestine. Okay, so now we have that. All right, let's see what they're asking us now, what to do here. And they said, which label parts correctly shows where absorption of water can occur? And I said always occur, but can occur. Now, the large intestine is the main place for water to be reabsorbed, right? So absorption of water will take place mainly in the large intestine. However, absorption also takes place in the small intestine. Water is not the main thing, but water can definitely be absorbed there as well. Okay. All right. So now let's look at where we see O and N. And so O and N will be our option B. Okay. So option B will be our correct answer here. All right. So let's go to the next question, which is question number 28. Okay, so question number 28 now reads that 1 gram of fat contains 37.0 kilojoules of calories. A slice of bacon contains 3.5 grams of fat. Now we say, how many calories will be in 2 slices of bacon? Now, first thing, let's just really calculate this real quick, yeah? And so, the first thing we need to know here is that if we talk about 2 slices of bacon, 1 slice is 3.5, right? And so 2 slices is going to multiply it by 2, and here you'll get 7, okay? You're going to get 7 here. But they ask us now the total calorie. In 1 gram of fat, we have 37 kilojoules. So what we're going to do now is to multiply this 7 by 37, yeah, by 37, and that will give us 259. That will be the correct answer here. So 259 will be the correct answer. All right. All right, great. All right, so 259. And so here we'll have D will be an option here. All right, so great. All right. Excellent. Now let's move on to the next question here. So question number 29 is said that the diagram shows the movement of food in the esophagus. Okay. All right. So the movement of food in the esophagus, and the question asks, what is the process called? So this is now called a wave-like movement. So here you have a wave-like movement. All right. So wave-like movement. So what I want to point out here is that the muscles will be contracting and relaxing and cause this wave-like movement of food along the alimentary canal. And so this part here is the esophagus. And so this process here is called peristalsis. So D will be our answer for this question. Now let's jump to the next question here, number 30. And in this one also, I've gone ahead and label for you as well. You can just study the labelings and know the different parts, but I'm going to point out a few things here. Now we said, which structure changes shape to allow gases in and out of the leaf? And so this one here, question number 30, will be D, because the guard cells will either open or they will what? Close. All right, so D will be your answer. Now what I want to point out here, a few things quickly, is that when you talk about the cell and the plant in the leaf and some specific areas, I just want to make note of them. So for example, this here, which is called the palisade mesophyll cells. Notice I put the palisade mesophyll cells as B. So the entire region we call the palisade mesophyll layer, okay? As well as a spongy mesophyll cell. One of them is a spongy mesophyll cell, and so the entire section we'll call a spongy mesophyll layer, okay? And so just to point that out, the vascular bundle will contain xylem and phloem. And so xylem is always on the top, phloem is at the bottom. That is inside of the leaf, okay? Inside of the leaf, the xylem will be more to the top, the phloem will be to the bottom, okay? And there's a reason for that, all right? Because the xylem will be closer to the cells that are photosynthesizing the most, which are the palisade mesophyll cells, all right? But they also will get to the spongy mesophyll cell as well. And the spongy mesophyll cell, notice they are spacious and they are more circular. And the reason why they're spacious is to allow gas to pass through easily, or to allow gas to diffuse through the leaf easily. All right, question number 31. Now, question number 31 is a diagram shows flower P and flower Q, okay? And so flower P and flower Q, they are different. So notice the difference between these flower. In flower P, the anther is out of the flower itself, okay? And so these will be the stigma showing right here. So these will be the stigma that I'm circling right now. In flower Q, the anther is pretty much close to the stigma. They are inside of the flower technically, all right? Or they're upright. So these are further apart. That's the major difference right there. All right, so let's now talk about this. It's a which row correctly states features of flower P and Q. All right, so look at the difference between them and you'll see what more likely will happen here. Now we're talking about petals and talk about pollens. So let's go back to the diagram. Since we talk about petals and pollens, what we're looking for is probably the type of pollination that takes place. And so if you look at the design of the flower, this type of flower will be more insect pollinated because the stigma and the anther, they are pretty much close. But in this one, flower P, they are far apart, okay? They are very far. As a matter of fact, the anther is outside of a flower. So if anther is outside of a flower, the flower will not attract any insect. So this will be like, for example, wind pollinated type of flower, right? All right, so let's now jump into it real quick. All right, so this is question number 31. All right, let's go back a little bit. All right, so let's go through it. And in this now, we said that P is brightly colored. Remember, the one that is insect is P, is wind is P, And so brightly colored could not be the answer for this flower at all. Could not be brightly colored. For option B, it said that P is dull, which is the wind pollinated plant or flower, and Q said that large sticky pollen, which is the correct answer here, because yes, dull, because if you're wind pollinated, you don't need to be brightly colored, and the pollen must be sticky if it's going to be insect pollinated. Okay, so large and sticky could not be work for P, and of course net like stigma could not work here. Net like could work for P, so I'm gonna stick that, but dull could not work for Q because it must be brightly colored. Okay, all right, so let's take the one that's actually correct here. So this is incorrect. This make this choice incorrect. All right, this also make this incorrect, so both of them are incorrect. Let's see, large sticky, no, because that is by wind. That's not correct. This is also correct right here, brightly colored. So Q could be brightly colored and also large sticky. All right, and this will be dull. So those would be the correct and the incorrect options for both flower. So you can always just kind of go back, all right, and you could see the different flowers and observe them carefully and know which one is which one. All right, let's jump into question number 33. And the question number 33, again, I've gone and labeled this these for you to make life easier and make us spend less time doing this. Now, so which picture shows a method of vegetative propagation that involves the use of rhizome? And so option A will be your correct answer right here. And so a potato, this is a potato, so it's a tuber, potato tuber. C is a corm, and then we have D, which would be runners. Okay. All right, so let's go to question number 34. So question number 34 said plant in different habitats have become modified to reduce transpiration rate. It said which correctly matches the plant with its adaptation. Okay, and so we have a cactus here, and so cactus broad leaves, which is not correct because cactus, if you think about cactus, a cactus, the leaves are narrow. In fact, they don't really have true leaves, really. Okay, and so B, it said mangroves, many stomata on leaf surface, and if you're going to reduce transpiration, then it can't have a lot of stomata because a lot of stomata mean more transpiration, right? All right, so the pine with needle-like leaves, and that's good because the leaves are needle-like. They're not really true leaves, but they're needle-like. And needle-like mean that they're very thin and narrow. That means the surface area is very small surface area, so not a lot of transpiration will take place. So this is a good one here for question number 34. So C look good. Let's see what D is saying. Right, rhizome, rolled narrow leaves. Okay, not true, but yeah, this one is the best adaptation right here. All right, excellent. Now 35. So question number 35 said the following experiment was set up to investigate the process of transpiration. It's a both sides of the leaves on the cut stem are covered in petroleum jelly. Now we have two conditions here. We have hot, humid day, and we have cold, dry day. Okay, and so we have plant R, S, T, and U. But let's make some note right here. The oil is used here for a reason, is to prevent evaporation of water from the beaker, and so therefore mean any water that is lost from the beaker must go through the plant. Okay, so the purpose of the oil is to prevent evaporation of water from the container or from the beaker, and so therefore water can only be lost from the plant or through the plant. Okay, all right, so now let's go to the question. It said which two beakers would show the greatest change in water levels? So the greatest change in water levels, so notice plant R and plant T, they are not covered with petroleum jelly, but plant S and plant U, they are covered with petroleum jelly, which means the stomata, they are blocked, hence you'll have less or no transpiration taking place in those two plants. And so R and T will be the one that will transpire the greatest, okay, which means they have greatest loss of water. What I just said just now, what do I say that? Okay, R and T. Okay, R and T will be the greatest. So this will be option B. All right, now let's jump into the next question here. And the next question here is, which is question number 36. And question number 36 is a diagram shows an extended human arm. All right, in interest of time, again, we have the bicep, which is the one that is before, and the tricep is the one that at the back of your arm. Okay, so you can say B before, so bicep is before, in front of your arm. Okay, and so he said, what combination will bend the arm? So if you want the arm to bend, bending of the arm. So how are you going to bend the arm? You're going to bring it upright or towards your chest area, correct? All right, so how to do that? I want to look at the muscles carefully. And so bringing this up, let me draw something here for you. So what you're going to do is to bring the arm like this, okay? Bring it up to bending the arm. All right, so the arm is going in that direction. So if the arm is going that direction, that means this muscle will be pulling this way, correct? And if that muscle is pulling this way, this muscle is pushing, will be pulled that way. So therefore mean that the bicep will get shorter, which means the biceps will contract, and the triceps, they will relax, which getting longer. So therefore means that this answer here is that the bicep getting shorter, which is contracting. The triceps, they will get longer, which is relaxing. And so option B seems to be pretty good. Okay, tricep relax, get longer, bicep going in, so they're getting shorter. Okay, so B will be your answer for that question. Now, question number 37, and again, really to make life easier for us, I'm going to label this, and just to point out why I label it in this way. Okay, I want to make a note right here. Okay, this will be dendrites and this will be nerve endings. Now, what's the difference between dendrites and nerve endings? Okay, is that you notice a clue right here, matter of fact. Dendrites always pick up the impulse and send it along the axon, and it will terminate or end at the nerve endings, and it will move to another neuron or move to the effector, right? And so importantly, this here is a sensory neuron, by the way, because the cell body is at the side, okay? And so this is a sensory, and so the end of this neuron, it will go into the relay neuron, okay? All right, so pointed out that the dendrites will pick up the impulse. So which structure has the function of picking up impulse or stimulus? That is the dendrites. That area is important. All right, so let's kind of label that, then move it out of the way, so I could label that one. So A will be your option here. All right. Again, I have the myelin sheath, and the myelin sheath will be the part that will insulate the neuron and also speed up the transmission of impulse. The cell body as a regular cell that contains a nucleus. We have the axon, which is the entire region here. Okay, and so just make a note of the labelings for a neuron. Now, question number 38. They said diagram shows a front view of the human eye. It said, what are the number structures? So what are the numbered structures? So what is 1, 2, 3, and 4? All right, right off the bat, you should know that number 1, right, is the center of the eye, so that's going to be pupil, while 2 going to be what? The iris. So you can look out for 1 and 2, iris and pupil and iris respectively. So let's go into this. So number 1 is A is saying cornea and eyelid, so that no, that's not correct. Looking for what? 1 is pupil, the center of the eye, that small opening, and number 2 will be the iris. So we know that pupil and iris here is correct for C, and then number 3 will be the sclera, and the last part of it, number 4, will be the eyelid. Okay, so we know that C will be your option right there. All right, so what you can always do, again, is to look at what you know, obviously, and then you look at what will fall into place after that. Is a way to eliminate the incorrect answers from the correct answers. All right, so this is question number, let me see, what question is this? Question number 39. And so question number 39 now reads that which of the following groups of symptoms most clearly suggest oversecretion of thyroxine in an adult? So thyroxine is important for metabolic rate. Okay, so it controls the metabolic rate. So if you produce a lot of thyroxine, then you're going to have a high metabolic rate, and therefore your chemical reaction will be much faster, And if you're producing, if your reactions are faster, you produce more energy, for example, because respiration process in the cells will be much faster, because metabolic refers to chemical reaction, okay? And so chemical reaction in the body, and so producing more energy, then the person will be more active. And if you're more active, then you tend to lose a lot of weight because you'll be burning your calories, you'll be burning your fat molecules, and the body just burning energy constantly, okay? And so therefore 39 here will be B. All right. All right, so let's jump into number 40. So number 40, it reads that which sex hormones, I'll go back to it a little bit. It says which sex hormones are responsible for bringing about puberty in males and females, okay? So in males and females, in a male, in a female, that's female on the first column. So estrogen is responsible, so the answer must be between B and C. And for male is testosterone, so 40 has to be B, okay? All right. Number 41, it said what is fertilization and where does it take place in humans? So fertilization is when the egg and the sperm cell, when they fuse, and this takes place in the fallopian tube. So it's a combination of gametes or sex cells, sperms and egg, and it takes place in the fallopian tube. So looking through, you know, option B is our answer here for question number 41. Now for question number 42, it said the HIV virus attacks the cells of which body system? And so again, the virus really attack the immune system because the immune system will be responsible to produce white blood cells that will fight against the disease or the virus. So this will be option C. Now for question number 43, it said the diagram below shows how an experiment was set up to investigate the condition needed for germination. Looking for the conditions needed for germination. And remember, I've talked about this about 3 or 4 times before. And germination, remember, does not require any light, neither does it require any carbon dioxide. So what we need, we must have available water, we must have the correct temperature, okay? We must have oxygen, and in some cases, the pH is also considered to be important, okay? So again, here now, let's go through each of them. We have WX, Y, and so look into each container. So W is at room temperature. The beans are soaked in water, and so the cotton here is moist. So we have moist cotton hull, and X, we have the beans are soaked, moist cotton hull as just the same, but the temperature is different. So we have 0 degrees Celsius. In Y, unsoaked beans, so there's something different with Y here. So the unsoaked bean, that is a little difference here. Let me just kind of highlight that. So the beans here are unsoaked, all right, and then the cotton hull here is dry. So this one is dry, unsoaked, and this is at room temperature. All right, so let's see now what they're asking us about this, these seeds and germination. It said which list shows the order in which seeds will germinate from the least to the most. All right, so let's point it out right here. Least to the most. So if you look here carefully, you'd realize now that if the seeds are dry, which means, and even the cotton hull is dry, then therefore mean there's no water available for the seed to germinate. So this will be the least one. So this will be number 1. And let's look between these two. They're the same exact setup. Soak seeds, moist cotton hull, moist cotton hull. What's the difference is the temperature. Now we have room temperature and we have 0 degrees Celsius. Now I'm going to point this out that most enzymes are inactive at 0 degrees Celsius, right? And so room temperature will provide a more suitable temperature for germination to take place. Now I want to make this clear that you have some seeds, however, will germinate in colder temperature. Typically not 0 degrees Celsius, because that is freezing. Okay, and so the moisture within the seed will be ice, will turn ice. Okay, and so therefore germination will not take place. However, though, some seeds require very cool temperature, not cold as 0 degrees, but cool. Okay, and that's why some seeds may germinate in a refrigerator rather than germinating outside of the refrigerator because of that cool temperature, and also the moisture present in the refrigerator as well. So from the least to the most, again, we're going to start with least will be our Y because this is dry, and then X, and then W. So we're going backwards. X, Y, X, and W. Okay, Y, X, W. Let's go for that. Y, X, and W. That is option D. All right. So let's go to our next question here, which is question number 44. All right, so question number 44 reads now, so which body cell is capable of differentiation into any cell type? And so not egg cell, not nerve cell, not sperm cell, but stem cell. Stem cells are specific in terms of forming many different types of cell. All right, so the next part here, next question, it said, the table shows the mass of four substances in 1 dm cube of blood plasma and urine. It said use the table to answer question 45 and 46. So question 45 and 46 now. Let's see what they're saying here. Before I go to the question, let's just read this real quick, yeah? All right, so here we have the substances. We have urea, we have sodium, we have protein, we have glucose. We have the mass in 1 dm cube of blood. So blood is on this column, and then we have urine on the other column. Sodium represents salt, okay? Because sodium can be found in salts. Just to bear it in mind. All right, so question 45 said, which substance shows the biggest mass, the biggest change in mass per dm cube between blood plasma and urine? So let's look at which substance will change the greatest, all right, or the biggest difference. And so by looking at the chart, you'll see clearly that 80 and 0.1 must be the greatest change. That's the most obvious answer. It's pretty obvious, a matter of fact, I will say, right? The rest, not so close as 80 to 0.1. That's a big difference, right? All right, and so let's say if you should minus them, what you get here is 79.9, right? And so that's a big difference. The others are way less. All right, so here will be our protein. So 45 will be C. So protein will be the biggest change. Now, question number 46 now reads, it said, what is the percentage decrease for sodium between blood plasma and urine? So let's look at the percentage change between sodium. So let's go to the chart and do a calculation right here. All right, so looking for sodium, the percentage change. So the percentage change here will be the difference between these two. So let's find the difference between those two. And so the difference between these two here will be 8.5. Let's type it in. 8.5 minus 4.6. Okay, and that will give us 3.9. 3.9, okay, and so we get 3.9 as a difference, but that's for percentage change. So for the percentage change will be 3.9 divided by the original, which is the mass of the thing in water, because they minus both, so it'll be 3.9 over 8.5, and this now will give us our answer here, which will be 45.88. Okay, and so this is 45.88 is our answer. So let's go down and see what we get here. So 45.88 for this option, and so 45.88 can be rounded off to 45.9. So option B is our correct answer here, okay, for question number 46. Now for 47, let's go to question number 47. Okay, question number 47. Now question number 47 reads that in tobacco plants, the diploid number is 48. Diploid means 2, so di means 2, and so diploid mean the 2 sets of chromosomes, which mean the full number of chromosomes. Okay, that's what diploid means. And then it said now how many chromosomes will be found in the nucleus of a pollen grain from a tobacco plant. So pollen now will be a gamete, which means gametes are produced by meiosis, and meiosis produce half the number of chromosomes. So half of 48 will be 24. So 47 is B. Okay. So half of 48 is 24. Meiosis produces half the number of chromosomes in each of the daughter cells. And remember, meiosis also produces 4 daughter cells. Okay, just to put that in. Now, question number 48 is which term describes the physical appearance of an organism? Straight off the bat, we know that this here is our phenotype. Phenotype shows the physical trait, the physical characteristics, or the observable traits, the things that you can observe about an organism. That's the phenotype. And please remember that phenotype is determined by the genotype. Genotype determines the phenotype, okay? So based on the genotype, you'll know the phenotype. Okay, great. Now, 49 now it reads that in peas, in pea plants, the allele for round seeds, which is uppercase R, is dominant to wrinkle seeds, which is lowercase r. It's in a genetic cross between 2 heterozygous plants, what percentage of the offspring will have round seeds? Now, I said this to you already before, okay? I hope you remember it. Is that when you have 2 heterozygous parents, the offspring ratio in terms of phenotype is always 3:1, which means 75% will be of the dominant trait and 25% will be of the recessive trait. Also, it's important to also note, right, that in terms of the genotype ratio is always 1:2:1. All right, and that's something you need to remember. Now, I'm going to work it out for you if you do not understand this, but you can look at the answer and know that, hey, this is 75%, as I said, because it is a 3:1 ratio. But for the benefit of those who might not understand how to do this, what you want to do is to do your genetic cross by drawing a Punnett square. And so the Punnett square look like this. And then in the Punnett square, you want to put your offspring. So what you want to do, put the parents on the outside. Now, heterozygous means that you have an uppercase R and a lowercase r. Why I said R? Because R is used in this example, okay? So based on the allele that you're using, then heterozygous means a dominant allele and a recessive allele. In other words, a dominant uppercase letter and a lowercase letter. Hetero means different, okay? Homo will mean the same. So homozygous will mean the same exact case of letters to use. So on one side, I'm going to put uppercase R right here. I'm going to put lowercase r right here. Okay, and so on this side, I'm going to put uppercase R as well because both parents are heterozygous. They said that a cross between 2 heterozygous plants. So here I'm going to get 2 uppercase R's. Okay, and remember, always use the same type of letter for the same trait. In other words, I'm saying if we're going to use, talk about the shape of the seed, then you use R, for example, not R and W to say W is wrinkle. Do not do that. Use the same letters or the same type of letter for the same trait. And so this is uppercase R and a lowercase r. Here we have an uppercase R as well and a lowercase r. And we're going by row by column. And here now we have 2 lowercase r's. So in this case here, just to point this out real fast, is that these 3 that I'm going to tick, let me tick those. As a matter of fact, let me just highlight those. The ones I'm going to highlight here, just put the highlight in these boxes. These will be our round seed because once it contains an uppercase R, the seed must be round because the dominant trait will always show, right? In the unshaded box here, which is a 2 lowercase, they will be wrinkled. A wrinkle, which is recessive, can only show when there are 2 recessive alleles, okay? And so out of the entire thing, 3 out of 4 will be round, which means 75%. 3 out of 4 times 100, 75%. So 75% of the seeds will definitely be round. Okay. last question, last question for the day. Now, it's the dog breeds that we have today were developed through which methods? And this was done by artificial selection. It was deliberately done by breeders to produce certain types of species. All right, and so this is where we close for today. And again, I appreciate you being here and trying your very best to make sure that you stay on top of the game in terms of passing your examination. And so stay blessed, stay safe, and look out for some other reviews. I'm coming with some paper 3s shortly. So until then, stay tuned.